This conference will now be recorded.
Good morning, one and all. Am I audible? Okay. So this is a revision class for the questions which were given to you in the exam. So I will be going according to the questions. So first important question is, so define apoptosis and mechanisms of involved in the apoptosis is a major question and is an important question in the first chapter. So as you all know, what is apoptosis? Apoptosis is a pathway of cell death that is induced by tightly regulated suicidal program. So it's a programmed cell death in which the cells are destined to die by activation of intrinsic enzymes which degrade the cell's own nuclear DNA and nuclear and cytoplasmic proteins. So it's a programmed cell death in which it is a suicidal program in which the cells die by the activation of the intrinsic enzymes. So these apoptotic cells, they break up into fragments known as apoptotic bodies. They contain portions of cytoplasm and nucleus. The structure of the cell is altered in such a way that these become tasty targets for the phagocytes. So there are two distinct pathways for apoptosis, and these main pathways they converge on caspase activation. So caspase activation is the major step involved in this apoptosis. The two major pathways are intrinsic pathway and extrinsic pathway. Intrinsic pathway also known as mitochondrial pathway, and whereas extrinsic pathway is also known as death receptor pathway. So intrinsic or mitochondrial pathway, it is a major mechanism of apoptosis on all mammalian cells. So mitochondria, these are the major important organelles which are involved in this apoptosis. These are organelles that contain proteins such as cytochrome C. So it results from increased permeability of mitochondrial outer membrane with release of pro-apoptotic molecules. Then the mitochondrial intermembrane space into the cytoplasm, and cytochrome C, when released into the cytoplasm, they initiate the suicide program or apoptosis. So you have to remember that mitochondria, which is the important organelle, it contains cytochrome C in its intermembrane space. When the cytochrome C is present within the mitochondria, the apoptosis does not occur. But when there is increased permeability of this mitochondrial membrane, and when due to any reason the cytochrome C is released into released out of the mitochondria, then the apoptosis is initiated. So the major mechanisms of apoptosis, one is the initial intrinsic pathway and the extrinsic pathway. Any injury like radiation, toxins, or free radicals, they cause effect on the mitochondria. These mitochondria, they have the regulators are, there are mainly two important types of apoptosis I will be telling later. So the release of these mitochondrial pro-apoptotic proteins, it is tightly controlled by BCL2 family of proteins. You have to remember BCL2 family proteins. This family is named after BCL2, which is frequently overexpressed due to chromosomal translocations. So there are more than 20 members of BCL2 family proteins, which can be divided into three major groups on their function. They are mainly pro-apoptotic, anti-apoptotic, and BH3 only proteins. So what are anti-apoptosis means? They inhibit the apoptosis. So like BCL2, BCLXL, and MCL1, these are the main anti-apoptotic proteins. They contain 4 BH domains. And coming to pro-apoptotic means that pro to apoptosis. They cause apoptosis of the cells like BAX and BAC. These are the major two prototypic membranes of this pro-apoptotic group. And like anti-apoptotic, they also has 4 BH domains. And the third group is BH3 only proteins. They act as sensors of cellular stress and damage, and they regulate the balance between the other two groups like pro-apoptotic and anti-apoptotic. These BH3 only proteins, they maintain the balance in between these two types of proteins, and thus acting as arbiters of apoptosis. So when there is a viable cells, the survival signal growth factor, there is production of anti-apoptotic proteins. Then the cytochrome C is maintained within the mitochondrial membrane. So there is no leakage of cytochrome C and the cell survival occurs. But when there is a lack of survival signal or any factors like irradiation or any damage to the cell occurs, there occurs DNA damage and activation of sensors like BH3 only proteins occurs and antagonism of BCL2 and BCL2 occurs. This leads to activation of VAX and VAC, which are pro-apoptotic, and then this causes leakage of cytochrome C from the mitochondria into the outside into the cytoplasm. This leakage of cytochrome C and other proteins leads to activation of caspases. So the major important step in apoptosis is activation of caspases, which is caused by leakage of cytochrome C from the mitochondria. So you have to remember that. So once released into the cytosol, this cytochrome C activates this combines with a protein called apoptosis activator factor 1, which forms this fusion of cytochrome C with the apoptosis activating factor 1 APF1 forms a wheel-like hexamer called as apoptosome. This complex is able to bind caspase 9, the critical initiator caspase of mitochondrial pathway, and the enzyme cleaves, setting up an auto-amplification process. So the complex of APF1 and cytochrome C is known as apoptosome. This apoptosome activates caspase 9. So this is the initiator caspase of apoptosis. Cleavage activates caspase 9, which triggers a cascade of caspase activation, thereby activating other pro-caspases, and the active enzymes mediate the execution phase of apoptosis. Then coming to the extrinsic or death receptor pathway, this pathway is initiated by the engagement of plasma membrane death receptors on a variety of cells. So mainly intrinsic pathway is due to release of cytochrome C from the mitochondria, whereas extrinsic pathway is by the activation of the death receptors. These death receptors are present on the every cell. On every cell there will be death receptors, but activation of these receptors causes in extrinsic pathway of this apoptosis. Okay. Death receptors are the membranes of TNF receptor family that contain a cytoplasmic domain which is involved in protein-protein interactions, and that is called the death domain because it is essential for delivering apoptotic signals. So this is the fast receptor. This is the fast receptor is the death receptor which is present on every cell, and this is a ligand known as fast ligand. When this fast ligand fuses with the fast receptor, the death domain is formed. This is known as fast associated death domain, FADD. So fusion of this fast receptor with fast ligand forms fast associated death domain. This fast associated death domain activates a inactive enzyme known as pro-caspase 8. This pro-caspase 8 is activated into active caspase 8. This active caspase 8 then in turn activates execution caspases, which leads to apoptosis. This autolytic caspase activation occurs from pro-caspase 8 to caspase 8. So this death receptor, that means activation of this fast ligand or fast receptor is responsible for the extrinsic pathway, and this is responsible for elimination of self-reactive lymphocytes and damage by cytotoxic T lymphocytes. So then coming to the summary, the mitochondrial pathway leads to activation of initial caspase 9, and the death receptor pathway to the initiation of caspase 8 and 9. So intrinsic pathway to activates caspase 9, and then extrinsic pathway to caspase 8 and 10. After the initiator caspase is cleaved to generate its active form, the enzyme death program is set into motion by rapid and sequential activation of the executional caspases. So major executional caspases are 3 and 6. They act mainly on the cellular components. So initiation caspases are 8, 9 and 10, whereas execution caspases are 3 and 6. These caspases, once activated, they cleave an inhibitor of cytoplasmic DNase, and thus make that this enzyme enzymatically active. This enzyme induces cleavage of this DNA, thus causing DNA damage. Okay, this is the major question, apoptosis. You have to remember mainly few major things are like apoptosis, the programmed cell death or suicidal cell death. In this, the cells are destined to die by the activation of these caspases. There are two major pathways. One is intrinsic pathway or extrinsic pathway. Intrinsic pathway also known as mitochondrial pathway, whereas extrinsic pathway also known as death receptor pathway. In intrinsic pathway, the release of cytochrome C from the mitochondria is the major important step. Any stimulus like radiation, DNA damage, free radicals, or any stimulus which causes increased permeability of this mitochondria causes release of this cytochrome C from the mitochondria into the cytoplasm, leads to activation of these caspases. Then, whereas in death receptor pathway, there is signaling to the death receptor which is present on every cell. Through the receptor it is mediated. So by the fusion of this receptor with the ligand, that domain is formed. This death domain in turn leads to activation of these caspases. So there are mainly two types of caspases. One is initiator caspases and the other one is executioner caspases. Initiator caspases are 8, 9 and 10. In this, caspase 9 is activated in intrinsic or mitochondrial pathway, whereas caspase 8 and 10 are activated in mitochondrial or in death receptor or extrinsic pathway. And the executioner caspases are mainly 3 and 6. So whatever may be the pathway, main step is this activation of these caspases, which in turn leads to the damage of the DNA and finally cell death. Okay, then coming to the second question, free radical mediation of the cell injury. So what are the free radicals? These are extremely unstable molecules which are highly reactive chemical species with a single unpaired electron in their outer orbit. So any species, any chemical species with a single unpaired electron in their outer orbit, they are known as free radicals. So in the cells, they attack and degrade nucleic acids, proteins, lipids, and carbohydrates. So they attack everything in the body and they cause damage. They initiate autolytic, autocatalytic reaction. That is, the molecules that react with free radicals, they are converted into free radicals. So any molecule which has paired electrons in their outer orbit also, when they come in contact with these free radicals, one electron will be lost and that become a single unpaired electron and then thus forms a free radical. So what are the main free radicals? Are mainly three types. One is hydroxyl radical, hydrogen peroxide, and superoxide. Okay, so they are the major free radicals. Free radicals, they constitute an important mechanism of cell injury. They mainly contribute to chemical and radiation injury, oxygen and other gases toxicity, cellular aging, microbial killing by phagocytic cells, and the inflammatory damage. So all these are mediated by the free radical injuries. So any radiation or toxics or perfusion, they cause production of free radicals like superoxide, hydrogen peroxide, and hydroxyl radical. These three are the major free radicals which are produced in the body. This superoxide free radical, it is converted to a hydrogen peroxide by the enzyme known as superoxide dismutase. So this hydrogen peroxide, it is also neutralized by preventing the damage to the cell death by decomposition to water by glutathione peroxidase or catalase. So these three are the major enzymes like superoxide dismutase, glutathione peroxidase, and catalase. These three are the major neutralizers of these free radicals. And this hydrogen peroxide, if this is not neutralized, it converts into hydroxyl radical. This is the free radical, and this free radical, it mainly causes lipid peroxidation, which causes membrane damage, and protein modification, which causes breakdown of these proteins and forming fragmented proteins, and finally DNA damage leads to mutation of these cells. So neutralization of these free radicals mainly occurs by vitamin E and A, which is present on the cell membranes, beta carotene, and within the cytosol, like enzymes like superoxide dismutase, catalase, glutathione peroxidase, vitamin C, and ferritin, cellular plasmin, these are present in the cytosol. And within the mitochondria, again enzymes like superoxide dismutase, glutathione peroxidase, catalase, peroxisomes. Within peroxisomes, catalase is present. These are the neutralizing factors of these free radicals. So when these enzymes acts on free radicals, they are neutralized and they prevent the damage of these cells. But in the absence of these enzymes, when free radicals are produced, they produce cell injury. So this hydroxyl radical, this converted into this superoxide, this converted into hydrogen peroxide by superoxide is prevented by superoxide dismutase. This hydrogen peroxide in presence of catalase is converted into water, and this hydrogen peroxide is a reversible reaction in formation of water in the presence of glutathione peroxidase. And in this process, only a reaction known as Fenton reaction occurs. In this, Fe plus 3 is converted into Fe plus 3, and this is the major reaction, that is Fenton reaction. So that is free radical injury of the cell injury. Then coming to the third question, define necrosis and enumerate various types of necrosis with examples. So what are the major patterns of acute cell injury, like irreversible injury or cell death? That is necrosis. Coming to the definition of necrosis, is the sequence of morphologic changes that follow cell death in living tissue. The sequence of morphologic changes that follow cell death in a living tissue. The morphologic appearance is mainly due to enzymatic digestion of the cell. So enzymatic digestion of the cell occurs in this necrosis, which is of two types mainly. One is autolysis and the other one is heterolysis. In autolysis, hydrolytic enzymes are derived from the dead cells themselves. That cell is death, and the cell death from the dead cell, these hydrolytic enzymes are released, and this causes the death of the other cells. Whereas in heterolysis, the hydrolytic enzymes are derived from the invading inflammatory cells. Okay, autolysis and heterolysis, and the other thing is denaturation of the proteins also occurs. Coming to the microscopic appearance of this necrotic dead cells, the mainly the what we see in under microscope are then cytoplasmic changes. The eosinophilia or pink is increased due to eosin binding to denatured proteins, and there is decreased basophilia or decreased bluing mainly due to imparted by DNA, and the glassy homogeneous cytoplasm is seen due to loss of glycogen, and calcification may occur in the late stages. These are the cytoplasmic changes which are seen, and coming to the nuclear changes due to the breakdown of the DNA, there are mainly three nuclear changes: the karyolysis, pycnosis, and karyorexis. Karyolysis is a decreased basophilia of chromatin or degradation of the chromatin. Pycnosis is a nuclear shrinkage and increased basophilia. Karyorexis is the fragmentation of this pycnosis nucleus. So this is a normal cell which contains rhizosome, nucleus, endoplasmic reticulum, and mitochondria. Whereas in a necrotic cell, the cell is swollen first. The rupture of lysosomes and the autolysis of this occurs. Nucleus becomes a shrinkage of the nucleus occurs, that is known as pycnosis. Then the karyolysis and karyorexis occurs. And mitochondria becomes swelled. Large densities of accumulation of in this mitochondria occurs. Defects in the cell membrane also occurs. Myelin figures are formed, and the lysis of endoplasmic reticulum. This is a picture showing a histopathology of the kidney with necrosis of the tubular cells. Then coming to the patterns of acute injury, there are specific morphologic patterns of necrosis. There are different types like coagulative necrosis, liquefactive necrosis, gangrenous, caseous, fat necrosis, and others like fibrinoid necrosis. The specific morphologic patterns in necrosis, coming to individual necrosis, one is coagulative necrosis. There is preservation of the structural outline of the dead or coagulated cells for days together. In initial days, there is preservation of the structural outline. The most common form of this is the most common form of necrosis, particularly in myocardium, liver, and kidney. The characteristic of hypoxic cell death in all tissues except in the brain. Myocardial infarction is very good example. The mechanism is denaturation of proteins and enzymes, which leads to blocking of cellular proteolysis, which leads to preservation of cell outline. So this is a mechanism involved in the coagulative necrosis. Coming to the morphology of this coagulative necrosis, grossly the tissue will be pale in color, normal firm texture at the beginning stages, and become soft later due to digestion by the macrophages, then which may lead to rupture of infarcted myocardium. Microscopically, in the first few hours, no abnormalities are seen, whereas in later, progressive loss of nuclear staining with preservation of cell boundaries are seen. Finally, at the last stage, damaged cells are removed by the macrophages. The presence of necrotic tissue usually evokes an inflammatory response followed by repair. So what is the fate of necrosis? Most of the necrotic tissue is removed by leukocyte by phagocytosis combined with extracellular enzymatic digestion. If necrotic tissue is not eliminated, it attracts calcium salts, which leads to dystrophic calcification. So this is the picture of coagulative necrosis. This cellular appearance looks like cooked, and the cell structures and delineation between the cell and cell is lost. And this is the picture of gangrenous necrosis. Liquefactive necrosis, the necrosed area, the field shows cystic spaces. These are cystic spaces surrounded by necrotic cell debris. Mainly liquefaction necrosis is seen in brain, and by the surrounding area shows granulation tissue and the gliosis. Caseous necrosis, The pink homogeneous material mainly seen in tuberculosis, and the fibrinoid necrosis seen in major vessels, mainly arteries, and seen the large vessels. This fibrinoid type of necrosis is seen. And there's a fat necrosis usually seen in liver. Foamy macrophages are present. Necrosis of the fatty tissue occurs. So this is mainly necrosis. You have to remember the difference between apoptosis and necrosis also. In apoptosis, the cells will be shrinkage of the cells will be there, whereas in necrosis, the cells will be swollen. Whereas in necrosis, surrounding inflammatory response is there, will be there, whereas in apoptosis, no inflammatory response will be there. The cell death occurs in both the cases, whereas in necrosis, the cells which die, they cause inflammatory surrounding inflammatory response, and phagocytosis occurs. In apoptosis also phagocytosis of the cell death occurs, but there will be no surrounding inflammatory response. Then coming to calcification. So what is pathologic calcification? Pathologic calcification is the abnormal tissue deposition of the calcium salts together with smaller amounts of iron, magnesium, and minerals and other mineral salts. So it's the abnormal tissue deposition of the calcium along with smaller amounts of iron, magnesium, and other minerals. So pathologic calcification implies the abnormal deposition of calcium salts in the soft tissue. There are two major types of pathologic calcification. One is dystrophic calcification and the other one is metastatic calcification. So coming to there are two general phases. One is initiation phase and the other one is propagation phase. In the initiation phase it might occur either extracellularly or intracellularly. Extracellular initiation occurs in the membrane-bound vesicles derived from dead or dying cells that concentrate calcium by their affinity for acidic phospholipids. Phosphates accumulate as a result of action of membrane-bound phosphatases. In initiation phase, the cycle of calcium and phosphate binding is repeated, eventually producing a deposit. Initiation of intracellular calcification occurs in mitochondria of dead or dying cells. Propagation phase of crystal formation mainly depends upon the concentration of the calcium and phosphatases. The structural components of the extracellular matrix such as collagen as well as other matrix proteins such as osteonectin and gamma carboxyglutamic acid containing proteins are also present. Coming to the dystrophic calcification, it is encountered in areas of necrosis where they are coagulative, caseous, or liquefactive type, and in process of enzymatic necrosis of fat. In these cases, dystrophic calcification occurs. It occurs in non-viable or dying tissues in the presence of normal calcium serum levels. Remember, dystrophic means disease, death. Dystrophic means mainly dead. They occurs in dead cells, mainly dead tissues, non-viable or dying tissues in the presence of normal calcium cells. It occurs in arteries, in atherosclerosis, in damaged heart valves, and in cirrhosis. Calcium can be extracellular. Whatever may be the site of the deposition, the calcium salts mainly appear grossly or macroscopically as fine white granules or clumps, often felt as gritty deposits, and histologically with usual HLD stain or these calcium salts have basophilic, amorphous, granular, sometimes appearance. They can be intracellular, extracellular, or in both locations. In the course of time, heterotrophic bone may be formed. On occasion, single necrotic cells have crystals that become encrusted by the mineral deposits, and with acquisition of outer layers may create lamellated configurations called Samama bodies. The name is given due to the resemblance to the grains of the sand. These Samama bodies, they are typically described as concentric lamellated calcified spherules. They are concentric with lamellated configurations. They are calcified. They are concentric lamellated calcified spherules. Sometimes types of cancers like papillary cancer, carcinoma of thyroid, they are apt to develop Samama bodies. Any malignancies which are mainly papillary type develop Samama bodies. In astrocytosis, calcified iron cells gather about long slender spicules in the lymph node. Tuberculous lymph node is virtually converted to a stone. This is a picture showing Samama bodies. This is a concept. Please come up below all of you. Then this is the picture of asbestosis, which are a long, slender, giving a dumbbell appearance. Composition of this asbestosis. Calcification of calcium. Then coming to the metastatic calcification, it is a deposition of calcium. Dystrophic occurs in death or dying tissue. Metastatic calcification occurs in tissue which is associated with hypercalcemia. Whereas in dystrophic there will be normal serum calcium levels, whereas in metastatic there will be increased calcium levels. There are four principles of metastatic calcification, mainly are increased secretion of parathyroid hormone like parathyroid tumors or ectopic secretion of parathyroid hormone by malignant tumors, destruction of bone tissue as in multiple myeloma or by diffuse skeletal metastasis, and vitamin D intoxication, renal failure causing secondary hyperparathyroidism. So these are the major principles of metastatic calcification. This may occur widely throughout the body, but principally affects mainly the interstitial tissues of the gastric mucosa, kidneys, lungs, systemic arteries, and the pulmonary veins. So this is the calcification, mainly two types, dystrophic and metastasis. Metastatic, dystrophic occurs in death or dying tissues. D for D. Metastatic occurs in viable tissues. Okay. Then coming to the fatty chain. In this fatty chain, you have to mainly know about the intracellular accumulations mainly. What is intracellular accumulation? This accumulation of any substance which is produced in abnormal amounts or normal substance which is produced in abnormal amount or any abnormal substance which is produced. Accumulation within the cell is known as intracellular accumulation. Within the cell it may be accumulate either within the cytoplasm or within the nucleus or both. So anywhere but within the cell, that is known as intracellular accumulation. There are four mechanisms of intracellular accumulation. The first one is inadequate removal of normal substance. So normal substance, when it is produced in normal amount or in increased amount, when it must be removed from the cell. But when the metabolism is inadequate to remove it from the cell, accumulation occurs, like in fatty changes in the liver. Then accumulation of abnormal endogenous substance, when there is abnormal production of abnormal substance within the cell, like when there is a mutated gene, there is abnormal folding of the protein occurs. This abnormal protein folding leads to inability to degrade the abnormal protein. This abnormal protein is not degraded, and which leads to accumulation of these abnormal proteins within the cell, like enzyme like with alpha-1 antitrypsin deficiency. Then coming to the third type, where failure to degrade by enzyme deficiency, like normal endogenous substances, when there is enzyme is produced, metabolite is produced, but the enzyme is absent, accumulation occurs like storage disorders. Any storage disorders. Then accumulation of exogenous substances, like exogenous substances when there will be no enzymatic machinery for its degradation or transport, which leads to accumulation, like a pigment. Any pigment which comes from the outside and accumulates within the cell, that is the accumulation of carbon in the lungs. This is the fourth type. These are four mechanisms of intracellular accumulation. First, coming to the lipid accumulation, this leads to fatty changes in the liver. Abnormal lipid accumulation in the cell occurs. It may be either triglycerides or cholesterol. Steatosis, or also known as fatty change or metamorphosis. Abnormal accumulation of triglycerides within the parenchymal cells occurs. Most common site is the liver, and it may also occur in heart, muscles, and kidney. Toxins, protein malnutrition, diabetes mellitus, obesity, anorexia, and alcohol are the major factors. So in normal lipids, which are free fatty acids from adipose tissue or ingested food, these are transported to the hepatocytes. These free fatty acids, when they are transported to the hepatocytes, they are either esterified to triglycerides or converted to cholesterol or phospholipids or oxidized to ketone bodies. So this is a normal metabolism of lipids. But when this metabolism is failed, the accumulation of the lipids within the hepatocytes occurs. So like triglycerides excess in the liver causing increased fatty acid entry into the liver cells, or excess production of fatty acid from acetate, reduced oxidation of fatty acids, or increased retention of triglycerides caused by inadequate complexing of this triglyceride with apoprotein molecules to form lipoproteins, or decreased release of lipoproteins from the hepatocytes. So in any of these causes, they leads to accumulation of lipids within the hepatocytes. So there are two patterns of the fatty changes in the liver. One is microvesicular fatty change. In this, tiny vesicles or microvesicles are seen, and the other one is large droplet fatty change. This is a Ray's syndrome. In this, multiple microvesicular steatosis and extracellular vacuoles with wide separation of the cells is seen. So when the normal cell, this is a normal cell, when there is abnormal metabolism or abnormal removal of the substance or lipids from the hepatocytes, the fatty liver occurs. Then coming to the very short questions, there are two questions. That is one, first one is metaplasia. So what is metaplasia? Metaplasia is the reversible change. Reversible change in which one differentiated cell type, that is epithelial or mesenchymal, is replaced by the another cell type. So one cell type is replaced by the another cell type, that is known as metaplasia. That is differentiated cell types. It often represents an adaptive response in which one cell type that is sensitive to a particular stress is replaced by another cell type that is better able to withstand the adverse environment. So when there is a stress to the epithelium it is unable to cope up with that stress, it is modified or it is replaced by another epithelium which can cope up with that stress. So this is mainly an adaptive response of the epithelium or any mesenchymal tissue. So what are the mechanisms of metaplasia? So metaplasia does not result from a change in the phenotype of an already differentiated cell type. Instead, it is the result of a reprogramming of stem cells that are known to exist in normal tissues or of undifferentiated mesenchymal cells present in the connective tissue. The most common epithelial metaplasia is columnar to squamous epithelium. So, example, in the habitual cigarette smoker, the normal ciliated columnar epithelial cells of the trachea and bronchi are often replaced by stratified squamous epithelium. So, bronchi are mainly lined by ciliated columnar epithelium, but whereas in chronic smokers or habitual cigarette smokers, this ciliated epithelium is unable to cope up with that smoking with that stress. So, it is replaced by the stratified squamous epithelium. The other examples are stones in the excretory ducts of the salivary glands, pancreas, or bile ducts, which are normally lined by secretory columnar epithelium, may also lead to squamous metaplasia by stratified squamous epithelium. And the third type is a deficiency of vitamin D or retinoic acid induces squamous metaplasia in the respiratory epithelium. So this is the columnar epithelium which is replaced by the stratified squamous epithelium. So the other type is this is a metaplasia of columnar to squamous epithelium. This is a columnar epithelium. This is replaced by stratified squamous epithelium here. The influences that predispose to metaplasia, if persistent, that is the influence if they persist continuously, they can initiate malignant transformation in the metaplastic epithelium. So thus a common form of cancer in the respiratory tract is composed of squamous cells which can arise in areas where the normal columnar epithelium has been replaced by squamous epithelium. So this is columnar epithelium which under stress is replaced by stratified squamous epithelium. But if the stress continues to be there, which is persistent, then the malignancy can arise from this squamous epithelium. So this is a common form of the malignancy in the lung. So metaplasia from squamous to columnar type may also occur in esophagus, which is known as Barrett's esophagus, in which the esophagus squamous epithelium is replaced by intestinal type columnar cells under the influence of reflux gastric acid. So the Barrett's esophagus is normally lined by squamous epithelium. Thus the squamous epithelium is replaced by intestinal type columnar epithelium, which has goblet cells. Okay. Squamous epithelium replaced by intestinal type columnar epithelium, that is known as Barrett's esophagus. Then coming to the connective tissue, metaplasia is a formation of cartilage, bone or adipose tissue, mesenchymal tissues in tissues that normally do not contain these elements. For example, bone formation in muscle designated by myocytic ossificans. So bone formation within the muscle, seen in myocytic ossificans. Okay. Then coming to the last question, that is lipofuscin. So what is lipofuscin? Also known as lipochrome or wear and tear or aging pigment. It is composed of polymers of lipids and phospholipids which are complex with proteins. So lipofuscin, also known as lipochrome or wear and tear pigment or aging pigment, composed of polymers of lipids and phospholipids, which are complex with proteins. It is derived through lipid peroxidation and of polyunsaturated lipids of subcellular membranes. It is a sign of free radical injury. So this is a myocardium, mainly showing a lipofuscin, mainly seen in elderly. This is the aging pigment. Okay, so these are the questions which are given to you in the last exam. So only I have discussed briefly only the questions which are given to you, and the rest of the things will be taken in the next class. So in the next class you will be having this exam of the second chapter, that is inflammation, and again revision of those questions also will be done. Okay, thank you for listening and all the best for the next exam. Thank you.